Good afternoon. I'm Shyam Tallury. I'm the USDA peanut curator in Griffin. And the other two names you see on this presentation are my colleagues. Miley Mobley is the technician in my program, and Dr. Melanie Harrison is our research leader. And some of you probably know that the US National Peanut Collection is maintained in a gene bank in at the Plant Genetic Resources Conservation Unit in Griffin. And before I start my talk, I thought I would show you all a slide of some terms that I'm going to use in my presentation, and so that you all understand what each of these terms mean, when you hear me use them during my talk, actually. So my title already has plant germplasm or peanut germplasm. So what is germplasm? Germplasm is any living tissue from which new plants can be grown. Oftentimes it's the seed, as in the case of peanut, but it could also be a vegetative stem cutting, which you can root it and then grow into a new plant. Or it could be a tuber that could give rise to a new plant. And it could also be like meristem or even an embryo that you can put them on an artificial nutrient medium to raise into new plants. So any living tissue from which you can grow a plant is what is germplasm. And if you just look underneath there, the next one is genetic or germplasm resources, which means any genetic material of actual or potential value. For example, an accession of peanut could be resistant to a particular disease. So that's the potential value. And we just heard about nematode resistance in stenosperma and other multiple disease resistances coming in from Betizacea and stenosperma in Dr. Birdie Early's talk and Carolina's talk. So those are the germplasm resources. They have the genetic material of potential value. So the other term that you hear me use during my talk is genetic diversity. Genetic diversity, as we know, refers to the total variability that's present within a species, actually. So oftentimes, genetic diversity is viewed as a way for populations to adapt to changing climates or changing environments, actually. So it's believed that genetic diversity, and I'll talk more about it in my following slides, provides some kind of buffer, like a buffer for to withstand abnormalities, actually. And so the last one is here, a gene bank. I said, like, the National Peanut Collection is preserved in the gene bank in Griffin, or a seed bank is a repository of all these living plant materials to conserve this genetic diversity that's present in all these germplasm resources. And my title also has a term, genetic vulnerability. So what I mean for this term is the susceptibility of a crop due to its genetic makeup. So again, I'll talk more about it in my later slides. So the goals of my talk is to demonstrate the importance of peanut, this genetic resources, or the diversity available in these peanut genetic resources, and the need for these germplasm resources maintenance or conservation. And the other aspect is to highlight the potential vulnerability issues for sustained peanut production in the US. So again, going back to genetic diversity and germplasm resources, as I mentioned already, genetic diversity is deemed essential for crop improvement, actually, for peanut improvement. And it's believed that genetic diversity lends resilience to crops against any future threats. And as such, it's important to preserve these genetic resources or germplasm resources. They must be managed well and are made available for cultivar developments. And I just want to emphasize here that the diversity of germplasm resources provides an insurance or some kind of, like I said, buffer for unforeseen threats or future threats. But I also want to emphasize that genetic diversity per se probably doesn't offer like an insurance against any future threats. It's the useful genetic diversity that's more important. As we heard in the previous talks, that's the stenosperma or the pteridocoi or other wild species accessions have the genes for resistance. That's the useful genetic diversity. A lot of times there is diversity, but it may not be really useful in a breeding program. So, and genetic vulnerability is real and must be addressed for sustained peanut production. And we heard this morning in the virus, the symposium, Dr. Culbreth gave a beautiful narration of tomato spotted wilt virus and the source of resistance in Georgia green via southern runner with the PI 203396. So I'm not going to spend much time on that. So, but it is a real issue. And those of us who have observed this or experienced this back in the late 90s and early 2000s with the tomato spotted wilt virus severity in the southeastern production. So again, Georgia green was the only variety at that time that had the field tolerance to this virus. And the resistance came again from this PI 203396, which was maintained or which is being maintained in the Griffin collection. So talking about the peanut germplasm resources, the National Plant Germplasm System, which is managed by the USDA-ARS Research Service, maintains this germplasm collection in Griffin and distributes the germplasm resources for research and education uses. And as you see in the picture here, there is tremendous amount of genetic or phenotypic variation for pod shape and pod sizes and seed coat colors and seed size. And these germplasm resources represent most of the global peanut genetic diversity. And we have, I believe, about 100 countries actually represented in this germplasm collection that we have in Griffin. So it represents a pretty large global genetic diversity and is the primary source for domestic as well as international research community for peanut improvement. And these germplasm resources provide, again, as I said, have several useful traits. They provided several useful traits for peanut improvement. And many of the breeding programs in the US have used these germplasm resources to develop high-yielding, climate-resilient cultivars for domestic production. And the germplasm collection in Griffin contains approximately about 9,500 accessions of both cultivated and wild species, and mostly preserved as seeds. Of course, there are some wild species in section Rhizomatosae and Tallury, which propagate by rhizomes or stolons. But for majority of the part, the wild species as well as the cultivated peanut collection is maintained as seeds in Griffin. And as you see in the top, all the wild species are regenerated periodically in the greenhouses. And the cultivated peanuts are regenerated in field plots to replenish fresh seeds into the gene bank collection. And we maintain the seeds at minus 18 freezers, actually, as you see in the top right. The germplasm is also backed up at the National Laboratory for Genetic Resources Preservation in Fort Collins. And that's the bottom right picture, actually. That's from Fort Collins, where the entire National Plant Germplasm System is being maintained, all the germplasm, not just the peanuts. So the peanut breeding activities in the US are mostly in the public sector. So there are several active peanut breeding and genetics and genomics programs at state universities, as well as at the USDA-ARS facilities in Tifton, Georgia, and in Stillwater, Oklahoma. And so most of these programs are responsible for the cultivars that are in the market now. And there are two other private seed companies, one located in Georgia and the other in Texas, also release cultivars for commercial production. So now moving on to the genetic vulnerabilities, threats, and limitations facing peanut. Again, as we heard this morning in the virus symposium and in Dr. Bertioli's talk this afternoon, the most commonly viewed threat is any evolving disease or an emerging pathogen or a pest, actually. So that's the most common. And right now for peanuts, the peanut smut fungus is the most urgent threat to focus on. And if people who are interested to read or learn more about this, Dr. Kelly Chamberlain has a poster during this meeting. So I encourage you to go and look at that and read. And the other thing, as Dr. Sudeep Bagh mentioned this morning again, the peanut clump virus and Indian peanut clump virus. These two viruses, or APRES has listed them as peanut quarantine pests in a couple of years ago. So none of these pathogens, either the smut fungus or the seed-borne viruses, are not present in the US peanut production regions right now. But that's why, but they are in like Argentina and in the Indian subcontinent and in Western Africa. So we got to be, again, proactive, as it was mentioned in this morning. So we got to be proactive, and we have already, like as I said, good news here is the source of smut resistance have been identified in collaboration with the international partners in Argentina. And again, I cited the reference of Dr. Chamberlain here and Dr. Dylan Vaughn published in Peanut Science last year. So the source of smut resistance have been identified. But the limitation right now is the lack of rapid, quick, and inexpensive and reliable diagnostic tools, actually. So again, the USDA-ARS group in Oklahoma is working on this to develop the diagnostic tools for smut resistance, as well as Dr. Sudeep Bagh has been developing the molecular diagnostic methods for the detection of these two seed-borne viruses.
We're under 2 minutes, Shyam.
Okay, I'll hurry through these. The genetic vulnerability, I mentioned here, like in 2019, the certified seed production included 21 public cultivars, actually. So most of the times, the certified seed production acreage reflects into what's being grown the following year. So if you look at it, in Southeast production, Georgia 06G, which was released in 2006, has been the most dominant cultivar. And similarly, in the Virginia VC production area, Virginia Carolinas, Bailey, which was released in 2010, has been the most dominant variety. So they've been in cultivation for quite some years now. And furthermore, in the Southeastern production, most of the other cultivars too, in addition to Georgia 06G, they have a lineage going back to C99R actually, and it's well known, like in the southeastern production, C99R derivatives have been predominant actually. So the vulnerability risk could be the dominance of just a few cultivars in an extended area of cultivation for extended periods. So the other issue is do we know the genetic diversity status of the existing cultivars? I believe there is insufficient knowledge of the genetic diversity within the US germplasm collection and commercial cultivars, although the progress is being made with molecular characterization of this peanut core and the African peanut germplasm collection. See, other issues I have highlighted here are inconsistent international pest pathogen monitoring systems. We heard this morning there's a need for a global surveillance system to monitor these new pests and pathogens. And the last two here relate to our inability to acquire and add new germplasm as the native habitats are being threatened, or the international treaty restrictions limit our ability to bring them in. So my recommendations here are, this is real genetic vulnerability, and diversity per se does not provide protection for future threats. It should be useful genetic diversity. And inadvertent introduction of pathogens through international trade is a possibility and is a concern. And as I said, global surveillance system with better coordination is needed. And so the cultivar development efforts must be focused on deepening the genetic base of cultivar gene pool. Again, as we heard from the previous talks, wild species offer that resource or any unadapted germplasm with useful traits. So there's a need for a concerted global effort, such as the one for peanut smut, needed for germplasm evaluation to identify sources of useful traits. So with that, I'll end. If there's any time, I'm happy to answer any questions. Thank you.